Welcome to Sanjana's Education Strategies. Learn the trade secrets to cover vast concepts in no time. If you like the video, please do like, share, and subscribe.
Hi students, so today we are going to start a new topic. So in this session we will be dealing with structure of chromosomes and also about chromosomes, then about DNA structure, replication of DNA, about the difference between DNA and RNA. So all these things we will be dealing in this topic. So I hope you like this video a lot and please don't forget to like, share and subscribe. Let's enter into the video. Cell, this is a cell, okay, this is a cell. Within a cell you will be having nucleus. These are chromatids or chromatin fibers. This is nucleus. Yes, okay. Now what happens to this chromatids or chromatid fibers means they start to condense. When they are condensing, you can see this is chromatid fiber 1, 2, 3, 4, 5. Okay, so one more and 6. So one chromatid fiber, it gets attached with another one chromatid fiber 2. Another one 3 attaches with 4, 5 attaches with 6, 7 attaches with 8. Like this it gets attached. Chromatin fibers attaches in this form, where in the center of their structure. So the center region where they are getting attached, that region is known as centromere. See how they will be formed after attaching. See, this is not a single line. Actually, I told you I'll be telling you about what a chromosome contains. A chromosome contains nucleotides. So many nucleotides will be present in this region, like this piles of histones. Okay, like this it will be present. So it will be somewhat thicker only. So this is the structure of chromosomes. About chromosomes, to say, they inside chromosomes only all the genetical processes take place. So chromosome, when you take, it is the carriers of genes. Genes, they are the carriers of heredity. Heredity. Okay. So all this happens inside chromosome only. Inside chromosome we are having genes. Inside gene we will be having nucleic acids. Nucleic acids. What are the nucleic acids? Nucleic acids are two. One is RNA and the other one is DNA. Okay. All this will be present inside this chromosome only. Where this chromosome is present? Chromosome is present inside the nucleus. Where nucleus is present inside the cell. Yes? Okay. So this thing you have to understand. Now, normally in animal cell, okay, and plant cell, there will be different mitosis. The division will be different. About all those differences I will be teaching in next video. Okay, this is a nucleus. Inside nucleus we are having chromatin fibers, or you can simply call it as chromatids. Okay, anything. And these chromatin fibers, okay, they develop, they condense and they form chromosome. This is a chromosome. I'll show you how. Just think. Okay. Within a nucleus, there will be many chromatic fibers. They assemble together. One, another one. One, another one. Again one, another one. See, two of them, they join like this. In the middle region, they join. Then, what happens is, in this region, they are joining and they are centromeres. This region. Okay, this region is known as centromeres. They are joining in the centromere region. Okay, so this is how chromatic fibers develop into chromosomes. Now we'll see inside chromosome what is present. When you take chromosome, it is made up of many strands of nucleotides. These are known as nucleotides. nucleotides. So how these nucleotides will be arranged means like this. Nearby they will be arranged. Okay, so when I take one nucleotide, I am naming this as 1, 2, 3, 4. Okay, like this. When I am taking first nucleotide, it will be arranged in piles. Can you see? This is first number, this is second number. First number, it is having many piles below it, like we arrange books, one below the other. Like that, in the form of piles they are present. Okay, and they are known as histones. Each pile, this is one pile, okay, this is pile A, this is pile B, this is pile C, this is pile D. This is from first nucleotide. First nucleotide, it is having A, B, C, D as piles. And this A alone, it is known as histone. We call it as A histone, then B histone, C histone, D histone. Now you understand? So histones together they form one nucleotide. Nucleotides together they form chromosome. Yes, that's it about histone. Now, when you take histone, one pile of histone we can just enlarge in the form of DNA or RNA. So this is DNA structure. DNA is double stranded. This is one strand. This is one strand. This is another one strand. Okay, this is another one strand. Two strands. That's why it's known as DNA is known as double stranded. Yes, got DNA. And you can see inside this DNA, what all these? Nucleotides, okay, arranged. So in one nucleotide, many DNA strands will be present. Now, in this DNA, we are having many things present, like first you are having sugar, or you can call it as pentose. Then you are having phosphate. Then you are having bases. That is, you can call it as nitrogenous bases. Nitrogenous bases. Now, you can just see this diagram. So, circle I have denoted for phosphate, then sugar, it's a pentagon, then base, it's a rectangle. Now, just listen how they are connected, you can see. If this is a strand, I showed you how the strand is, like this, right? So, in this strand, they are connected like this. Now, listen, phosphate is present, it will be pairing with sugar. Again, sugar pairs with the second phosphate, again it pairs with sugar. Like that, in both the borders it is happening. Now in the center region, you have to keep in mind bases. All the nitrogenous bases will be present in the center. Now here you have to know what are the nitrogenous bases. Okay, you are having 4 nitrogenous bases. One is adenine, another one is thymine, next cytosine, next is guanine. Yes, these are the 4 nitrogenous bases. Okay, in RNA, thymine will be replaced by uracil. In RNA alone, thymine will be replaced by uracil. Yes, in DNA structure, we will tell about thymine only. Now we speak about thymine, forget about uracil. Yes. Now, just listen. Bases connected in the middle. Okay. Now, we'll see how these bases are connected. Have you understood? Rectangular base. Adenine, it pairs with thymine. Guanine, it pairs with cytosine. This thing you have to learn. ATGC. Okay. ATGC. Adenine pairs with thymine. Okay. And guanine pairs with cytosine. Adenine pairs with thymine with double bond. Guanine pairs with cytosine with triple bond. And here you have to understand this bond is hydrogen bond. bond is hydrogen bond. Okay, this bond is hydrogen bond. That thing alone you have to understand. Okay, in this way pairing will take place. ATGC, then ATGC. Like that the pairing will take place. It may be AT or it may be TA. Anyway, but A must pair with only T. Adenine must pair with only thymine. Cytosine must pair with only guanine. Yes, this is the thing which you have to keep in mind. So in this way it is arranged. Phosphate and sugar will be connected. Okay, everywhere. Pentose sugar. We'll just call it pentose sugar, not simply sugar. Okay, and in the middle region, this is how nitrogenous bases will be present, and they are connected, or they are forming a bond, hydrogen bond. Yes, that's it about the structure of DNA. About female and male. Okay, so how many number of chromosomes are present in female and male? So I told you already, normally in a human being there are 23 pairs of chromosome. In that last pair, that is 23rd pair, okay, it is sex chromosomes. Now we are going to see about that sex chromosomes. In female we are having X and X chromosome. In male we will be having X and Y chromosome. That is one X, when you take it will have 23 chromosome, another one X it will have 23 chromosomes. So totally we will be having 46. For male also 46 chromosomes. When you add, totally we will be having 46 chromosomes. Now the baby born. Okay, that is when fusion of male and female gametes take place, zygote will be formed. Okay, and that zygote develops into baby. All the stages we will be learning in reproduction chapter. So that baby, okay, that child will have either XX pair or XY pair. Either females X and males X will join and form XX. If it is forming XX, then that child will be female. Okay, and it will also have totally 46 chromosome, that is 23. Okay. Then if females X and males Y is combining, then the baby formed will be male. Okay, and that baby will have 23 chromosome and 23 chromosome. Again it will retain 46 number. So here what they are coming to tell is, female it will be having 2n number, male also will be having 2n number. N. Only the n part, 1. 12 means n plus n, right? So only one n will be retained by the child. From one parent, only one n will be retained by the child, by the baby. Like that, from male parent also one n will be retained. So totally it will also get 2n number, like the parents. Like the parents, baby also will be getting 2n number. So this is about the sex chromosomes. Okay, now we are going to see about the replication of DNA. So this is a double-stranded DNA. So what happens is, this double-stranded, it splits. Okay, you can see how it splits. This is one strand, strand A. This is strand B. Okay, A and B. So it splits, and this A strand, A single strand, it produces a copy of that strand. This is a duplicate copy formed by strand A. Okay, and this is another duplicate copy formed by strand B. Both forms duplicate copies of its own strand. Now this duplicate copy splits the complete DNA into two, and it forms two daughter DNA. Now two DNA is formed. Okay, this is how replication of DNA takes place. Okay, this is, okay, see, one DNA it is having that parent A strand, another DNA will be having the parent B strand. But both this A and B will not be retained by one daughter DNA. It will be separate only. This is two daughter DNA. This is a red color indicates its duplicate strand. Okay, it's duplicate, it's copy. So that's it about today's topic. If you have any doubt, you can please show it in your comments, okay, so that I can understand what is your doubt and I can clarify your doubts in my next video. So please keep supporting my video and don't forget to like, share and subscribe. See you soon.